Hello everyone, this is the second lesson in second year syllabus, sexual reproduction in flowering plants. I will just make a quick revision of this. What have you studied in this lesson? Structure of flower. I am sure you can recognize parts of flower. Well, there can't be any direct question based on that. Structure of anther, components of anther. Anther wall, different layers of anther wall, sporogenous tissue. Similarly, structure of ovule, parts of ovule. Development of male gametophyte, development of female gametophyte. Different stages in development of gametophyte. That is pre-reproductive event, pre-reproductive structure and event. And then fertilization, double fertilization. Before that, you also study pollination mechanism. Different examples, including pollen-pistil interaction, compatibility, outbreeding devices, etc. And then you go to that embryogenesis, development of embryo from zygote, development of embryo in monocot, dicot. Initial stages are similar, but at the end you can differentiate monocot embryo from that dicot embryo. Differences between this endospermic seed and non-endospermic seed. Different types of fruits, examples, and you also focus that apomixis, polyembryony, examples. So it's easy for you to recall this quickly by taking NCERT summary or exercise questions or practice papers, etc. I will use few more terms to focus certain things very clearly in this lesson. Archisporium. This term doesn't appear in any paragraph. You have to go to summary part. Why did I take this term? How is it relevant to reproductive structures of flower? Yes, archisporium gives rise to megaspore mother cell or embryo sac. That will become embryo sac later. Pollen sac is also derived from archisporium. So both male and female reproductive structures of flower are derived from primitive tissue called archisporium. Polygonum. This is one important example. It's missing in your textbook. You study anatropous ovule. It was first described in the species Polygonum. You have studied a monosporic development. Yes, this shows monosporic development because there can be bisporic, etc. But heterosporic, that is out of NCERT. But you have studied monosporic. Most common mechanism is monosporic, polygonum type. 7 cells, 8 nuclei. I am talking about embryo sac. That's also called polygonum type. So this is like the species which was initially studied in flowering plant. Epiblast. Where do you find this labeling? In monocot embryo, there is small reduced second cotyledon. That is epiblast. There is scutellum. I am not talking about that. Second reduced cotyledon is epiblast. The explanation is missing in the paragraph. Entomophily. You study pollination mechanism. Pollination takes place through water. Pollination takes place through wind. Pollination takes place through animal species. When it takes place through water, it's hydrophily. When it takes place through wind, it's anemophily. When it takes place through animal species, it's zoophily. It can be ornithophily, keratophily, entomophily. You should be able to identify such flowers. Color by looking at the color or nectar or pollen, etc. Unitegmic. This word is missing in this lesson, but there is a sentence. Ovule is covered by 1 or 2 integument. They don't say 1 or more, 1 or 2. In angiosperm, ovule has 2 integument. Cycas has unitegmic ovule. It has only 1 integument. So it's necessary because you study cycas in plant kingdom. It's necessary to connect those examples. Navaschin discovered double fertilization. That is very unique in angiosperms. You know it. Apospory. That's development of gametophyte from sporophyte without meiosis. That is very strange. There is no meiosis. It's like apomixis. In apomixis, there is seed development without fruit, without syngamy. It's aposporic. Protogyny, development of gynoecium before the development of androecium. What is the advantage of this type of adaptations in plant? Which plant shows this condition? So it's necessary to collect this type of information. Protogyny is the characteristic feature of this Annona, Michaelia. The development of gynoecium takes place before dehiscence, before the pollens are released. It means when the pollens are released, the gynoecium will lose its viability or receptivity. The opposite condition is protandry in Malvaceae family, that's hibiscus. There is maturation of stamen or dehiscence, release of pollen before maturation of stigma, style or ovary. What is the significance of this? Once again, it's outbreeding device. There is no scope for self-pollination. The pollen has to come from some other plant, some other flower. There is only provision for cross-pollination. So there are such mechanisms. Heterostyly in passiflora. Stamens and carpels are at different levels. So there is less chance of transferring pollen into the stigma of the same flower. Okay. And we also have the examples of self-sterility. That's again outbreeding device. Pollen comes in contact with the stigma. Actually, it's a genetic mechanism. There is no chance of germination, no pollen-pistil interaction. In Solanum, there is self-sterility. Okay, there are many such, because you study outbreeding devices, but some of these terms are missing. I wrote here TZ, that was asked in one of the competitive exams. Tetrazolium chloride. It's used in seed viability testing. Farmers store seeds. In horticulture department or agriculture department, the people store seeds. After 1 year, 2 years, they would like to test viability, because after taking those things for cultivation purpose, they cannot have the conclusion that these seeds have lost their viability. So before introducing the seeds into farmer's land, this testing is mandatory. Viability testing used by, that is done by using this tetrazolium chloride. It becomes red colored formazon in the presence of dehydrogenase. It would be reduced. Imagine seed has lost viability. Seed doesn't show any respiration. No Krebs cycle, no dehydrogenase, no activity, no metabolism. There is no change in the color. Tetrazolium chloride will not become formazon. No red color. Seeds have lost the viability. So that is one of the important techniques in agriculture. Viability testing, seed technology. I said about already protogyny, self-sterility, heterostyly. Then by taking NCERT diagram, you have to look at few more additional types of ovules. NCERT gives you diagram of anatropous ovules. You have to list other type, orthotropous ovules, campylotropous ovules, cercinotropous ovules, with examples, types of ovules, types of endosperms, alluvial endosperm, nuclear endosperm, or cellular endosperms. So it's necessary to go through types, examples. Alluviae family has alluvial type, that's monocot. In many plants having gamopetalous condition, there is cellular endosperm. Coconut has free nuclear endosperms. So go back to that part. Bisporic, tetrasporic. You have studied monosporic only. What is bisporic? Two spores degenerate. Two spores develop into embryo sac. Embryo sac develops from two spores, bisporic. It is seen in Liliaceae, onion. This is seen in tetrasporic, is seen in peperomia. So 4 spores, all those 4 spores contribute to the development of embryo sac. Transmission tissue, that was also asked in one of the exams. It's the special type of tissue in style. Many pollens come in contact with stigma of one flower. Do all these pollens germinate in style they have to reach the ovule or embryo sac. This is the style between stigma and ovary. Do all pollen grains germinate simultaneously inside the style? Yes. That depends on number of ovules. Number of pollens germinate. There is transmission tissue. It is present in style. It's solid style, hollow style. There are different type of style. Many pollen tubes can germinate in a style through this special type of tissue, transmission tissue. And finally they reach embryo sac, synergate, where the egg is located. They can pass through salazogamy or integument, mesogamy, or directly they reach a micropyle region of the ovule, that's sporogamy. So we study that mechanism, mode of entry of pollen tube. I have got one simple direct question here. Which of these is wrongly matched? Orobanche, striga. You do find the examples of these two in the NCERT. Check. They are parasitic weeds. That is correctly given. Papavera. It's not apocarpus. It is syncarpus, multicarpellary syncarpus. This is wrongly given. This is correctly given. So you have to eliminate this. This is correctly given. You have to eliminate this. Endosperm mixed seed is also seen in onion, castor, onion. You can find the diagram in the textbook. But this is wrongly matched. I said multicarpellary syncarpus. It's not apocarpus. Thank you for watching.